Welcome back to the Neurostimulation Podcast. I'm your host, Dr. Michael Passmore, and today I'm joined by Dr. Osama Elyamani, a psychiatrist and neuroscientist whose work is helping to shape our understanding of transcranial alternating current stimulation, or tACS, as both a research tool and a potential therapeutic intervention in psychiatry. His group has published influential reviews and clinical research exploring how brain oscillations might be modulated to improve psychiatric outcomes. We'll be diving into the science, the clinical evidence, and where this field might be headed. So stay tuned. It's going to be a really interesting conversation, and I think you're really going to get a lot out of it. Dr. Elyamani, thanks so much for joining us today. We're really looking forward to this conversation and just really interested in your work, and I appreciate that you're taking the time out of your busy schedule to share all of this information with us today. So thanks very much.
Thank you very much for the invitation. I'm pretty excited actually to be with you at your YouTube channel, and it's really interesting to share the stuff with the audience. And hopefully we get a nice feedback.
Definitely, yeah. Thanks again. Maybe we can start by you providing a little introduction, yourself, your background, where you're located, and a little bit about what your lab is currently working on.
Yeah, with pleasure. My name is Osama Elyamani. I'm a postdoc. I have just finished my PhD dissertation. I'm pretty excited for sure to share this piece of information with you. Just finished it in July 2025. I'm working on neurostimulation, especially tACS and tMS in the context of psychiatric diseases, more specifically schizophrenia. And we try to record with EEG and MRI the activity of the brain and accordingly target a specific activity associated with psychiatric disorders and psychiatric symptoms and modulate them in order to normalize them, and hopefully we help patients with these disorders. I'm located right now in Germany, Liebig University in Gießen, and supervised by Professor Christoph Muhlert. He has a lot of work done in this field. Thank you.
Yeah, that's great. Well, it's really exciting to just kind of get involved in all of this, venture out and try and explore these ideas that you've got passion for and interest in. And I'm curious, in terms of that, what would you say first drew you to investigate this particular topic of brain oscillations and tACS, tMS in applications in neuropsychiatry?
Well, it's a very hard question to start with, to be honest. The point is, we try to find the fingerprints of the psychiatric disorders and the psychiatric symptoms. And as you might know, it's really limited in patients with schizophrenia or psychiatric disorders, or even in non-clinical populations. We don't have access to brains in comparison to rodents or monkeys whatsoever. So we have to depend on the status quo of methodologies or methods we have. Usually we have electrically or magnetically related methods like EMG, EEG, or imaging tools, MRI, and including for sure functional MRI. And if we look at these, we tend to have patterns of activities in the brain, either indirectly with MRI or directly with EEG. I'm recording the electrical activity of the brain. And that's why we have chosen to focus on the electrical activity, because it's a direct measure of what's happening in the brain, although it's really limited in terms of its spatial resolution, but it gives us a very good temporal resolution, and that's why we are interested in this. And our work is pretty related to the association of brain oscillations and how we could target these. Are they causally related to the different brain states? And in our case, it would be the psychiatric symptoms. And in that case, we could try to modulate them. And that's why we have chosen this pathway in order to prove this causality or this causal nature of the electrical activity that we could measure with EEG, for example, and to modulate them accordingly with stimulation techniques. And in our case, tACS. Why tACS? Because it tries to simulate the electrical activity of the brain, sending alternating current, alternating waves to simulate these coming from the brain that we measure, and hopefully we could modulate them in terms of potentiation or even decreasing them, and in order to modulate the corresponding functional or functions of the brain, and for sure the symptoms, which are main goal at the end of the day.
Yeah, no, thank you for that. That's a really succinct way of explaining a very complicated premise. So I think one of the key things, as I understand it, is the distinction between the spatial resolution that would be afforded by imaging techniques like MRI in particular, and then versus the temporal resolution, so the real-time brain changes that can be measured by EEG and how tACS influences it. Am I on the right track with that?
I think so. And even to add one more limitation, once we stimulate with tACS, we are not able anymore during the stimulation to record the electrical activity of the brain, because of the huge amount of artifacts that we induce in the brain. So we kind of depend on the EEG oscillations. The EEG measures oscillations, and we try to modulate them, but we can't measure the direct action or the direct mechanism of action, let's say, or the influence of tACS on the brain because of the artifacts. So we have to depend as well on MRI. On the other side, MRI gives us a huge or quite I would say robust spatial resolution. On the other side, we have this drawback of temporal resolution. We could try to combine both for sure, but still we have to have some kind of compromise. There are some papers actually which try to measure even the local field potential inside the brain, like intracranial recordings. I think this limited amount of papers gave us as well access to what's really happening in the brain electrically upon stimulation with tACS. Still, we could still try to use MRI in this regard using the hemodynamic change, which is an indirect proxy or indicator of brain activity. That's the limited world that we have right now.
Right. Yeah. And it makes sense to be getting the best of both worlds in terms of using the tools that allow for the spatial resolution and the tools that allow for the temporal resolution in order to get the best understanding. So I'll put an image up of a basic tACS setup, but perhaps you could just briefly explain for those who are less familiar with tACS compared with other clinically available technologies like TMS and tDCS. Do you mind just giving a brief overview of tACS and how that perhaps differs from those other two modalities?
So I would formulate it like this. tACS is more or less at its infancy in comparison to the other modalities. I think TMS was one of the first to be discovered and even used in clinical populations. It's FDA approved, I think, in Canada as well. In EU, it's quite approved as well, more specifically for depression. It sends magnetic fields in order to stimulate a specific parts in the brain. And one of the most used regions, dorsal prefrontal cortex, because it's quite related, a central hub for different connective functions in terms of depression. We could call it, or we could consider suprafacial stimulation. It could induce action potentials at the target brain region, dorsal prefrontal cortex. And with a specific timing, using specific stimulation protocols, we could induce synaptic plasticity that would lead to different consequences in the brain, to potentiate specific pathways in the brain that would lead to the relief of depressive symptoms. On the other hand, tDCS and tACS, they use electrical stimulation. So we put some kind of electrodes on the skull and we try to target specific brain region with these electrodes. But there is a huge difference in terms of both. tDCS, as the name would say, properly, some people might not know about the abbreviation. It's transcranial direct current stimulation. tACS is transcranial alternating current stimulation. So tACS sends kind of alternating current, it's oscillating current in terms of waves. tDCS is just sending at one electrode like positive activity and the other like negative activity. So it's kind of constantly inhibiting or stimulating one region of the brain. In comparison to tACS, and that's the uniqueness of this method, since alternating current, it simulates what's really happening in the brain. Our brain is really oscillating. It's all about rhythms, like different rhythms in different frequencies. And that's why we tended to tailor the stimulation frequency and the parameters using tACS in order to simulate these in the brain. And we drive with our own frequency what should happen in the brain. For sure, there are a lot of complexities that we try to overcome with this method, but the concept behind it is that we simulate what's really happening in the brain in comparison to tDCS with direct current constantly inhibiting or stimulating, and TMS with sending specific magnetic stimulation and causes action potentials. In comparison to TMS, both tDCS and tACS cannot induce direct action potentials. They increase or decrease accordingly the probability to fire action potentials in a specific phase. In terms of tACS, for sure, at the peak, the wave goes down, and so on and so forth. And I think this is quite important to consider. It will not induce this huge amount of stimulation at the target brain region, but we would induce a specific rhythm in the brain. I think this is like at the heart of it, what's really happening.
Yeah, no, thanks for explaining that. And that kind of fits with my understanding in terms of this important concept of the entrainment, or as you say, tailoring. I like that term, to try to entrain or tailor or encourage or prime, I suppose, the oscillations, and not only perhaps in terms of modulating brain function, but also in terms of, it's curious as well, this idea about it being a potential application on the diagnostic measurement side, as well as potentially on the therapeutic side as well. Yeah, it's really interesting. And I suppose the potential, or I guess what would you say the implications are, that has bearing on potential therapeutic properties in terms of things like neuroplasticity as well.
Yeah, I totally agree. I think you have highlighted a very important point that we have already mentioned in our systematic research review, in this review in the European Archives of Neuroscience. We try to encourage other scientists to work with this like an infant method, which is tACS, to discover how the brain would react to this oscillating current. The brain is sending on its own oscillatory waves, and if we simulate them, if we induce with our own rhythm, what would really happen? And this reactivity would really help us not only in maybe treating some diseases in the future, which we don't have at the moment with tACS, but also maybe for diagnostic purposes or even prognostic purposes, how the brain would react to this kind of modulatory action. I think maybe a subpopulation of patients might react differently, and accordingly we could try to diagnose them. This would be really amazing, because right now in most of the psychiatric disorders, or even all of them, we don't have objective measures. If we will ignore Alzheimer's disease, where we have amyloid plaques and tau protein, this whole story, in schizophrenia, depression, and the other guys, we don't have this neurobiological correlates. We do have a lot of evidence for sure. Like we try to find some associations, but we don't have this direct pathophysiology. I think brain oscillations might help decipher this picture and this reactivity to a tACS specifically, because it simulates again the brain activity, might be one way trying to discover this.
Yeah, so I guess on the diagnostic side, then the idea would be like as a biomarker kind of tool. Yeah,
sure. I think we are for sure limited with EEG because they tended to record the surface of the brain. We don't have huge amount of information from deep structures, so we're kind of limited, but we still have a direct proxy or indicator of the electrical activity of the brain. So we can depend on this, and the reactivity of this kind of activity again would be as well interesting way to look at the brain.
Yeah. Well, I was curious because in the review that you just mentioned, your team highlighted altered oscillations and the importance of, I guess, the oscillatory signature or something like that across a variety of disorders: schizophrenia, depression, OCD, ADHD, dementia, neurocognitive disorders. So I guess I'm curious, and maybe this is a bit too broad of a question, but which, if any, of those disorders do you think perhaps are the most prominent? So let me see, which of those disorders would you predict maybe are the most promising initial targets for tACS therapeutic interventions?
So you mean which diseases might show hope using this?
Yeah, I guess so. What may be the most promising? Maybe it's a difficult, we could go disorder by disorder, and then you could kind of, or if there's one in particular that you think lends itself best to this kind of technology, both in terms of the biomarker potential and or in terms of some therapeutic potential.
Okay, that's a very important question. I think before asking this question, we could ask ourselves a very other question, which is, do we think we have to approach psychiatry the typical or the classic way of doing it, in disease-based approach, or rather with symptom-based approach? If we do a symptom-based approach or kind of brain state-based approach, and given that some brain states are connected to or associated with the specific patterns and the electrical fingerprints, I think this would be kind of interesting. We don't deal with these diseases, depression as a whole, but rather depressive symptom or specific symptoms that might overlap actually in different, these kind of clinical pictures are kind of overlapping. And we have, for example, schizoaffective disorders, which are kind of like really overlapping with a huge umbrella of symptoms. So I think, especially with tACS, we might focus on specific brain states, specific associated with this brain state, and we target them according to what we record with EEG or MEEG. So I would say, I think in the future, most of the researcher might focus in this direction, like a symptom-based approach rather than a disease-based approach. If we will discover kind of electrical fingerprints related to the whole disease, so we could try to, for sure, like do with a classical disease-based approach. But I think what we know about the brain, that it works in different states, or it has situational, let's say, electrical fingerprints, and we could target these, especially if we could consider kind of like closed loop stimulation, that we, in real time, we target specific brain networks with the specific brain oscillations with tACS, for example. But back to your question, if I try to kind of predict in which direction or which disease would show most of the hope, I think it would be the disease that has specific brain networks involved, and we kind of discover them like the most. Something like depression, where we have specific networks implicated in the pathophysiology, or something like obsessive compulsive disorder, where we know like specific networks are kind of activated or inhibited, and they lead to the symptoms of OCD. These two diseases might be like interesting to target with the ACS.
Yeah, well, thanks very much for explaining that first part, because I think that's extremely important. And it was really interesting because in a recent episode with Doctors Hummel and Colzado, we talked a bit more about this idea about how neuromodulation technologies are helping to encourage people to rethink a little bit about how neuropsychiatric disorders are characterized in terms of the diagnostics, right? And perhaps not necessarily being beholden to this legacy, although it's important, obviously, for consistency of approach. And so in terms of medication approval, therapeutic approval, research studies, all this kind of stuff, obviously there's importance in having diagnostic criteria. And this is not to disrespect the legacy of the DSM and any of that stuff, but I think a lot of us who have clinical experience understand that there are shortcomings to that, and all the difficulty that goes into thinking about having to pigeonhole patients, so-called, into like specific diagnostic categories. So combining that, again, it's a matter of not either or, but getting the best of both, right? So combining that legacy diagnostic approach with, as you say, a symptom-based approach that fits more with these kinds of technologies and the biomarkers, the electrical signatures that go along with certain brain states and specific symptoms that cross in between different disorders. It's a really important point. Yeah, and I guess that also makes perfect sense in terms of those disorders specifically that seem to be correlated with particular network disruption. Yeah, it's fascinating, because I think part of it as well is that perhaps the disorders, that these other ones, the schizophrenia and then the dementias that seem to be perhaps, well, are all these disorders are complex, but the ones perhaps that are more complex than others in terms of like a global brain state disruption, or have multiple different symptom complexes, I mean, I think if we take something like schizophrenia, I suppose you could conceivably break it down into those. I think we're taught the three clusters: the positive symptoms, the negative symptoms, and the cognitive symptoms. Yeah, it would be interesting to know a little bit about whether tACS might be helpful in terms of modulating, because I think the medications are pretty good about helping to reduce positive symptoms, shall we say. Obviously, there are shortcomings and side effects, this and that. But really, there's historically been a real dearth of treatment options for the negative symptoms and the cognitive symptoms. Do you think perhaps that tACS might show some promise in terms of treating any particular symptoms of schizophrenia?
Okay, that's a very hard question, but especially talking about schizophrenia. Me personally, I think that schizophrenia could be, as you said, like kind of subclassified or subgrouped into sub-schizophrenia. We see a huge amount of variability in the clinical pictures, and in our own daily psychiatric activity, we are facing every single time a kind of new cluster of symptoms, let's say. And so it's kind of a syndrome, cluster of symptoms, and that's why might jump in to target these specific symptoms. And I think we are still, in the phase where we need a lot of research to be implemented. First of all, to subclassify these schizophrenias or to kind of discover them and for sure associate with them specific electrical fingerprints or signatures, as you said, and to target these very specifically and hoping to alleviate the symptoms. I would say in terms of this legacy problem, as you said, we don't have to consider medications versus stimulation techniques or all or none fashion or manner. We could combine both, where we consider antipsychotics kind of dirty drugs, where we have mostly a lot of receptors blocked or activated, deactivated, and so on and so forth. We fine tune the brain. They are neuromodulators somehow with the neurotransmitters being targeted, like dopamine, serotonin, whatsoever. Hopefully we can do it in the future, target dopamine in specific regions and serotonin in other regions and so on and so forth, but it's not the case. We do it unselectively in the whole brain. With the help of we could add this kind of specialization, this kind of specification of targeting. So we kind of tailor what we could target. So we could use both to dramatically, let's say, reduce the positive symptoms. It's very well known all around the world that are very, very important or play a very important role with positive symptoms, but with negative symptoms and cognitive symptoms, etc., domain, we sometimes even worsen the situation especially with the older. I think the tACS has now its role to jump in and try to figure out what really happens in the brain and to target specifically these networks. And I think because some of these cognitive symptoms and negative symptoms are being overlapping with some other disorders, what we said in terms of depression, so we could do kind of transdiagnostic approaches where we recruit patients with depression, schizophrenia, some other disorders, and all of them might share the same symptom, like working memory disturbance or cognitive disturbances or impairment, and we stimulate them with tACS. I think this could be kind of fascinating. So we go transdiagnostically and we find something that could target a specific symptom. I would not say that a specific stimulation would really improve the whole picture of a psychiatric disorder. This is like, I think from my side, is really naive story because we're targeting a specific brain network and we don't have this holy grail network in the brain that would affect the whole condition of patients.
Mm-hmm. Yeah, no, for sure. I think again, we've touched on this in similar conversations in previous episodes, but this whole idea of personalized treatment. So having the tACS as a potential option just in conjunction with medication and psychotherapy, both in terms of the diagnostics and then the therapeutics, which is fascinating as well. But I guess okay, well, let's maybe move away then from talking particularly about diagnostic categories, but if we talk about, say, a depressive syndrome, put it in those terms, it seems that there are some studies that show perhaps frontal alpha tACS in normalizing oscillations, improving symptoms, but it seems that those improvements are short-lived. But do you think that there's some kind of promise in terms of that type of an approach?
Yeah, I think so. I think so. To be honest, alpha has a lot to do with depression. There is this concept like frontal alpha asymmetry. There's no huge consensus in this regard. Some people could manage to prove it, some couldn't find it in the depression patients or the depression populations, let's say.
Mm.
I would say dorsolateral prefrontal cortex specifically has a lot to do in terms of depression. That's what we know from research, from MRI, from EEG techniques, but also from interventional approaches using DMTS, for example. So, and that's why a lot of researchers are pushing forward to using as well frontal tDCS to treat depression. I'm not sure about Canada, but I think in Europe it's really advancing and trying to get it approved or even off-label use and so on and so forth. I think, yeah, the frontal alpha would play an important role. I think there is a very famous lab, Fluffy Frohlich's lab in USA. I mean, they are doing a lot of research in this regard, and I think they are achieving quite a lot. And we have mentioned this. I think some of their papers in our systematic review. Again, it depends how you will tailor it. And I think you touched at a very important point in this regard. tACS would be helpful to tailor, to further personalize medicine in general, psychiatric approaches, because we target specific brain oscillations that are maybe unique for this specific patient or that patient. And we could even further personalize it, applying closed loop approaches. In real time, we apply a stimulation according to the actual oscillatory pattern of a specific patient. So I think, yeah, back to your question, it's really interesting to apply this in a personalized way for sure. For example, frontal alpha asymmetry or frontal activity in the brain, and DLPFC would be a very fascinating target for this.
Yeah, and I think thanks for explaining this concept about the importance of understanding the personalization piece and the individual differences in EEG activity, brain oscillations in different states in different individuals. It makes me think about how it's well known that experienced meditators are much more, shall we say, I don't know the specifics, but my general sense is that they maybe would have a much stronger propensity to have alpha states in general versus someone who doesn't, or that they have more control over that or something along those lines. So that might be an example of this type of, depending on someone's background and to the degree to which their symptoms maybe correlate with what their baseline oscillatory activity is, and all of that type of consideration.
Yeah, sure. And I think alpha is one of the main rhythms in the brain. We do see it like in EEG analysis and MEEG, where alpha is really prominent in our oscillatory pattern. Once we close the eyes, we do stimulate it. We do potentiate it, especially in the occipital activity. And I think the default mode network plays a major role in this regard. And as you said, even with meditation or even tiredness, we do see it in our laboratory on every single day. Once we record like resting EEG before and after a specific paradigm or mental activity, we do see this tiring effect, let's say.
Yeah. I'm curious. One of the courses I've been doing online is a recent course on TMS, actually. And in one of the later sessions, they talked about a study with tACS in post-stroke patients who have left hemi neglect. And that was really fascinating. And I think in general, I was understanding that there is some emerging evidence that tACS as a diagnostic kind of biomarker tool might be able to help differentiate between something like amnestic mild cognitive impairment and mild stage Alzheimer's disease, because that's a challenging differentiation often. And I know that it hinges on functional impairment, but then often there's a gray zone there. And obviously patients are anxious to know one way or the other. And the more we can give them as far as advice with biomarker tools, and hopefully non-invasive biomarker tools, because there's the LP and this and that, but if there are other non-invasive diagnostic tools, it would be very helpful. Do you see tACS as a promising technology to help with early stage diagnostics in neurocognitive disorders?
Yeah, I think so for sure, and I think you have already seen it. Like in one paper, tried to use this in the review we have published. But again, we will refer back to the nature, let's say, how brain oscillations are being developed or are developed in the brain. They depend on, or the oscillator, let's say, formulated in this way, the brain oscillations are coming from pyramidal neurons, more specifically from layer 5, let's say, in the cerebral cortex. And they're pretty connected. There is a huge connection in the brain. The whole brain works, depends on connectivity, the whole different brain regions. And even within each region, we have interneurons that tended to inhibit these pyramidal neurons. And at the end, if we consider this whole collection in a specific cerebral column or cortical column, we do see rhythms in the brain. So imagine if we lose a neurodegenerative disorder, some of these neurons or interneurons or whatsoever, I think the ability of this cortical columns or these cortical columns to produce the same oscillatory activity would differ, and it would be like really maybe compensating or decompensating. I think if we induce much more current from outside, like externally injecting current, and seeing the reactivity, how this internal rhythm would change upon a stimulation, this would give us kind of, I think, hint what's really happening in terms of neurodegeneration. This is how I see it. I look at like the very basis of these brain oscillations and what's really happening. We do know that these cells die in neurodegenerative disorders like Alzheimer or whatsoever. And once they die, I think we lose some of the rhythms that these neurons were responsible for. And the ability to react to externally injecting current, I think, would differ for sure. And building on that, I think we would use tACS maybe in the future to further diagnose how much we have of neurodegeneration, for example, or how much we have kind of impairment in the function of a specific brain region. That's how I approach it, let's say.
Yeah, no, I think that makes sense. I mean, and again, kind of going back to the distinction between spatial resolution and temporal resolution, perhaps it might make sense, because these days, of course, we would do, typically we might recommend a sequence of images in someone whose diagnosis is unclear, maybe an MRI at baseline, and then again, the following year or two, and then so on and so forth, to see if there's any progression in atrophy, I suppose, but then perhaps EEG could similarly be done at baseline, and then at year 2 and year 4, whatever, and in non-invasive again, and not particularly expensive, I suppose. And then that could be perhaps in some way combined with tACS as a way of figuring out, okay, well, is there a biomarker signal here that's concerning?
Yeah, for sure. And I think in this regard, I have to highlight something that at least we try to approach in our laboratory. There's a source localization to use EEG, brain scalp activity, and try to find what's really happening at the source level within the brain. We do have some methods trying to estimate the source activity, although they are pretty limited, but we could kind of connect in a very rough estimation what's really happening in every single gray matter source, hippocampus or whatsoever. I think they are quite limited. I will not highlight their very huge importance, but we could use these techniques, and maybe we could combine them as well with something like functional MRI, and we have 2 specific indicators, even from deep brain structures. Yeah, for sure, I do agree that we have a very limited spatial resolution, but if we use kind of MRI-based EEG analysis, this kind of, it would be, I would say, for the current state, like holy grail for the brain activities, that we have like both of them, like the spatial resolution from MRI and the temporal resolution from the EEG. I mean, that's the best of what we have right now, you'd say.
Yeah, and I have this vague recollection. I don't have any clinical experience with interpreting EEGs. I've never had that training, but I do recall something around frontal in particular, perhaps there being some intranasal electrode placement that might help to provide some specific measurement of frontal function. Is that something that maybe can be, or is being researched as part of the tACS protocol?
I don't know, to be honest, but I think, I mean, for sure, the more coverage we have over the head, the more we could measure the electrical activity of the brain. But what I could say here in this regard, that There are, for sure, again, some limited intracranial recordings, like in different papers already published. I mean, with epilepsy patients, like most of them. And again, we don't have physiologically acting brains. I mean, they're not functionally normal, to be honest. We are looking on the brains of epilepsy patients, which more or less would show some other dynamic from a non-epilepsy population. So I have never heard of it, to be honest, but I think that would be interesting for sure. I mean, like to have the more we cover the head with electrodes, for sure, the more we would get like a glimpse of what's really happening in the brain, especially if we use or apply later on source localization.
Yeah. Well, it strikes me just in reviewing the literature that in general, the safety and tolerability of tACS is strong, not really concerns about side effects or any particular risk of seizure induction or anything like that.
Yeah, indeed. I think tACS may be the safest or one of the safest neurostimulatory techniques. What we know that TMS, because it's a suprathreshold, as we said, a suprathreshold stimulation, it might unfortunately induce some epileptical shocks or whatsoever, or epileptical cases. If we do have the risk factors for this, we do screen for sure before applying it, but still there is a huge risk to development. And that's why we try to exclude patients or even participants within risk to develop epilepsy or convulsions on stimulation. tDCS, it's more or less safer than TMS, but still it has more adverse events compared to tACS. I think according to the current literature, tACS is the safest right now. Even for the fine tingling sensation and the local sensation, tACS is proven to be the safest. To my knowledge, or according to the best I know, there is no one epileptical case that was reported with tACS, for example. I think not to be biasing, one factor is that we do exclude in advance patients, participants who might have any risk to develop epilepsy. In a nutshell, it's much safer than tDCS for sure and TMS. Maybe because of this oscillatory pattern, because in tDCS, for example, we do have, again, subthreshold stimulation. We don't induce action potentials, but we do constantly induce or increase the probability of having action potential with this cathodal or nodal way of stimulation.
Yeah, no, thanks for explaining that. I think that's typically it makes sense that that's one of the major hurdles to overcome in order to get regulatory approval for therapeutic applications. So I'm curious to know, what would you see if you were to look forward into the next 5, 10, 15 years? Does it seem that based on your research and your expertise in this, would you hope that tACS will become more clinically available for biomarker diagnostics and or therapeutic applications, or both, I guess?
I'm not the very good person to predict what's really happening in the future, but anyway, tACS is still, again, it's at its infancy. We do have some clinical trials, even some registers on them. If we go to clinical trials, I forgot the website, clinicaltrials.gov, I think.
Okay, yeah, clinicaltrials.gov, that's right, yeah.
I mean, if we go to the website, clinicaltrials.gov, we do find a lot of clinical trials using tACS. I'm not pessimistic, but because we were still discovering how tACS is working and how to optimize the parameter, I think this is a very important aspect in tACS. We didn't reach the optimal conditions, or let's say, we didn't set the stage to further apply it with prominent hope to treat patients. But hopefully the other colleagues will really find some important results in clinical populations. But we do still have to apply very basic research to optimize the tACS parameters and to further discover how it's really working and how to maximize its effects. So I would say in the coming 5 years, I personally might exclude that it would be approved, unfortunately, because we do have a huge myriad of parameters to consider. There will be a lot of positive results in the literature, But again, it's about reproducibility and convince legalization institute to get it approved. I think it might not be convincing for that stage. In 10 years, for sure, we do have hope that we will do it, especially that it's quite practicable to apply it. It's really practical to compare it to TMS, for example. TMS should be applied with a specific device that's quite huge and it has its own coil and this technical hurdles, let's say. tACS on the other side could be the device and the technical equipment could be minimized. And I think there is a hope, or there is kind of hope for even home applicable stimulation. I think there is, for example, some companies in Europe, at least, which I have seen, they do provide home applicable electrical stimulatory devices for tDCS, and they are quite convenient. I have tried them on my own head, and I think this would be really comfortable for the patients to have something at home. And we could even connect it with the internet, and we have control over it as researchers or as clinicians. Something like this would be pushing us in this direction. But again, in 5 years would be really hard. In 10 years, I would assume so, hopefully.
Yeah, no, it's important to be hopeful about it. And I guess it's an exciting time, especially to be launching a research career and to be also combining that with a clinical career, and to be in a field that's in its infancy as well must be very exciting, because there's so many different potential opportunities and directions for exploring the science and this particular technology in terms of all of the possible therapeutic applications.
Yeah, indeed. And again, I would say, because it's really practical to apply it, I think this would encourage a lot of researchers and a lot of clinicians to apply it in the future.
Yeah, and then I guess maybe on a closing note, I'm just curious, is there any particular guidance or advice that you might give to students or early stage researchers, people who are working towards their PhDs, in terms of their direction, or what you might encourage them in terms of your own experience and where you see the field of neuromodulation in general heading? Any words of wisdom for them?
Oh, wow. That's a very hard question, but I would say I'm still as well like a postdoc, so I'm still as well in this phase or this stage. I would say we research on its own is really competitive for sure. It's really kind of hard to especially if you combine it with your clinical field. I mean, for me, for example, I'm a psychiatrist. I think the most important thing that to keep passion and to keep motivation, that you do something that you really like. I mean, don't jump in a PhD program or kind of, I don't know, field of research that you don't really like. I remember that I worked sometimes day and night in order to discover or trying to discover a very specific piece of result or whatsoever. If I didn't have the passion to do so, or the motivation to do so, I would not spend that time. And for sure, the second thing would be, we have to ask ourselves, because neuroscience is really, really branching, and there is a lot of multidisciplinary approaches and so on and so forth. We have to ask ourselves, I do categorize it in this way, which field you'd like to work in, because of like a lot of sub-fields in neuroscience, which method you'd like to use, and which organism, let's say, you'd like to work on. Because if you'd like to have some invasive, some more, let's say, access to the brain, you could consider something like rodents or monkeys, where you could invasively, for sure, with some limitations according to the ethical regulations, you could approach the brain. The methods you're using, for example, in humans, we do have some that, again, we have already discussed their limitations, like MRI and EEG. And the fourth factor would be the topic, what you'd like to discover. You'd like to work, for example, in my case, like auditory hallucinations, schizophrenia patients, or depression treatment, or just discovering the pathophysiology, and so on and so forth. So in a nutshell, you have to really, really spend a lot of time to choose the field that would grasp your motivation, and it would be depending on these four factors: the field you would like to work on, and methodology, organism, and for sure the very specific topic, of research.
Thanks so much. It's so clear that you're passionate about that, and I really appreciate you taking the time to share your story and your passion, your insights, and really helping us to navigate this exciting frontier in neurostimulation, helping us to understand tACS and all of these fascinating potential applications, all the way from the diagnostic biomarker side through to the personalized therapeutic side. It's clear to me from this conversation that tACS holds great promise for psychiatry, but it also raises important questions that will need ongoing, careful, rigorous investigation in the years ahead. As you say, it's in its infancy relative to some other treatments, but bringing all of these treatment and diagnostic options together to personalize the approach for patients is really, I think, going to be the key in the upcoming years. And so thank you again, Dr. Elyamani, for joining us. For viewers and listeners, I'm going to link Dr. Elyamani's team lab's key papers in the show notes. So I do encourage you to check those out. And please add comments or questions in the show note in comment section below, and I'd be happy to get back to you with that. And yeah, thanks again, Dr. Elyamani. Really appreciate the conversation and just wish you and your lab all the best in your research endeavors and in your clinical work also. Thanks so much.
Thank you very much for the invitation. I do thank you as well for your words. And maybe a last message from my side, feel free to drop us some messages. We don't bite if you'd like to consider the young researchers.
Fantastic. Yeah, that's great. It'll be wonderful to keep in touch, and hopefully we'll be able to meet at a future conference, something like that in the next months and years. So thanks again.
Thank you.
And so for viewers and listeners, thanks again for tuning in. Until next time, be well, stay curious, and join us next time as we discuss and explore new frontiers in the boundaries of neurostimulation and neuroscience. Thanks very much.